Okay, let's start then. So I wish to welcome everyone to this week's seminar here at Evolutionary Zoology and Human Biology Department at the University of Debrecen. Today's guest is Dr. Jente Ottenburghs from the Wageningen University in the Netherlands. He is the British Ornithological Union's Journal Policy Publicity Officer. So he is the person writing nice posts and the IB's journal blog at BOU. He obtained his PhD from Wageningen University, where he studied the genetic and hybridization of different geese species. He did his studies at Uppsala University in Sweden and returned as a lecturer to Wageningen University. He is the manager of the Avian Hybrids Project, a website that gathers all the available scientific literature on bird hybridization. So today's presentation will be also about hybridization, I presume. Jente, the floor is yours.
Yeah, thank you, Attila, for the nice introduction, and thanks for inviting me. It's really nice to kind of be in Hungary. I've been there a couple of times on holiday, and actually at home. So I'm from Belgium, and my family has a Wisla, so I have some connection with Hungary. You already nicely introduced me, but I always show this slide of the trajectory of my career so far. So I studied in Belgium, where I did my bachelor and master. Then I went to the Netherlands for a PhD, and then a postdoc in Sweden. And the funny thing is that this is exactly the migration route of a barnacle goose. So actually the logical route after my postdoc in Sweden would be to go to Russia, but I decided to migrate back to the Netherlands, and now I work in Wageningen. And today I'm going to talk about three main topics. So I'll start with hybridization in birds, to give you an overview and also some concepts that I will use later on in the talk. Then we dive into my favorite group of birds, the geese. So I'll show you some of the results from my PhD I did in Wageningen. And then we move to the Taiga and the Tundra Bean Geese as a kind of case study. And this is something that I worked on during my postdoc in Uppsala. So when we start with hybridization in birds, you can ask the question, well, how common is this? So how many species have hybridized? And a lot of people have tried to quantify this. So one of the earliest ones was Jente in France, who made a nice overview of the known hybrids at that time. Then in the 1950s, there was Annie Gray with a checklist. And the most recent one is the Handbook of Avian Hybrids of the World by Eugene McCarthy. And I used these data, including some newer records, to get an overview. And it turns out that if you look at all bird species, about 16% hybridize in the wild. And if you include captivity, it goes up to 22%. And what you see here is all the different bird orders. And the pie diagrams indicate the amount of hybridization. So in green, you have species that don't hybridize. In blue, the ones that hybridize in the wild, and then in red, the ones that hybridize in captivity. And the size of the pie chart indicates how many species there are in a different order. And what you can see is, for example, this one here. These are the water birds, waterfowl. So I'll come back to these. They hybridize a lot in the wild. And then here you see parrots. So they're quite popular to hybridize in captivity. And as Attila already mentioned, I run the Avian Hybrids Project, where I keep track of the literature and update the records of known hybrids. So this is a screenshot of the 4 latest blog posts that I wrote. So you can see it goes from woodpeckers to sparrows to phylogenies. So if you're interested in evolution of birds, you can check it out. But as I said, the Anseriformes or waterfowl, that's a kind of hybrid heaven. You get all kinds of crazy combinations. This is one of my favorites. If you cross a mute swan with the gray-leg goose, you get something called a swoosh. Here you see mallard and northern pintail. So you can see characteristics of both species. You see the characteristic tail of the pintail and the color of the mallard. And this is a more exotic one, a cross between a woodduck and a chestnut teal. And it's important to keep in mind that hybridization is not limited to just two species interbreeding. Often multiple species can interbreed with one another. And I quantified this for the waterfowl. So each dot in this network is a species, and the colors correspond to the genus. And if there's a line between two dots, it means a hybrid has been recorded. And you can see connections going in all directions. And if you then count these connections, you see that most species hybridize with just one other species, but a lot of them hybridize with multiple species. And then there's this one outlier, that's the mallard, which hybridizes with, at this count, 39 species, but it might even be more. Now, this is hybridization on a species level, where it seems to be quite common. If you look on an individual level, it seems less common. So as I told you, on a species level, it's 16%. If you look on an individual level, it's about 0.064%. So this was a study by Justin et al. Last year, and they used eBird, so an online website for collecting bird observations, and they counted the number of hybrids that were observed. So that's where they got the number from, and they compared it to an estimate by Ernst Mayr in the 1950s. He used museum specimens, and he had an even lower estimate of hybridization. So you can see, going from museum specimens to wild birds, there's quite an increase, and probably this number is even an underestimate. There might be even more hybrids that are missed because they're difficult to detect, or people just don't record them. And there's a nice discussion going on in the journal Evolution. So together with Dave Slager, I wrote a short commentary on this paper, and later on Justin et al. Did the same. So if you're interested in this discussion, you can check it out in Evolution. But what I wanted to focus on is, you see all these hybrids, but then the question pops up, why does one species choose a partner of another species? So for example, you have here a barnacle goose. Why would it choose a partner of a greater white-fronted goose and get the hybrid here in the middle? And when you look at geese, there's three mechanisms. And for the first one, I'll introduce you to these two species, the Hawaiian goose and the emperor goose. And these live in quite remote places. So the Hawaiian goose, as you can already guess, lives on Hawaii, whereas the emperor goose you can find in eastern Russia, on the islands between Russia and the US. So they're quite remote, but they're very popular in waterfowl collections. So people in the Netherlands that have a collection of water birds, they often have these species. And what happens is that they escape into the wild and they start looking for a mate. But of course, they cannot find one because all the Hawaiian geese are on Hawaii, all the emperor geese are somewhere in the east. So at the end of the breeding season, they settle for a partner from another species, and you can get hybrids. And this is known as the desperation hypothesis, because they get so desperate that they settle for another species. And the second mechanism has to do with nest parasitism. So some geese, they lay their eggs in the nest of another individual, hoping that they will take care of the young. But in some cases, the egg ends up in the nest of another species. And when the egg hatches, the gosling, so the young goose, will get imprinted on the foster parent, which is a different species. And later on, it will look for a partner of that species because of this wrong imprinting. And this seems to be quite a common mechanism in geese. And then we move to ducks, who have a more aggressive mating style. So this is the Argentine ruddy duck, which is known for having the longest penis of all duck species. And you can see that it's twisted at the end, like kind of a corkscrew. And the interesting thing is that in males, if the corkscrew is clockwise, the vagina of the female will be counterclockwise. It's a kind of defense mechanism against all the forced copulations by the males. So during the breeding season, they get very aggressive, and they mate with all the females that they encounter, even if they are a different species. So these forced copulations, they can lead to hybrids as well. But this seems to be more common in ducks than in geese, although it's also been observed in snow geese, for example. So these were the three main mechanisms when we look at geese. I'll now briefly introduce some concepts that I will use throughout the talk. And the first one is introgression, which is gene flow from one species into the other through hybridization. And what you see here is a nice graphic where you have a blue species and a red species. And when they hybridize, you get a hybrid that's 50-50. So 50% of its DNA is blue and 50% is red. And when a hybrid then back crosses with a red species, again, you get the 50-50 division. And you can see a small blue section has now moved into the red species. And the same has happened on the other side. So a hybrid has back crossed with the blue species, and a small part of red DNA now belongs to the blue species. So this is introgression, and it's quite common. A very famous example is between humans and Neanderthals. And what you can see here is my own genome. So I had my genome sequenced a couple of years ago. And a nice extra is that they also estimate the percentage of Neanderthal DNA in your genome. And it turns out I have 3%, which is above average. I have no idea what that means, but it's nice to know. Now, this introgression can have consequences. The exchanged DNA can be beneficial. And one of my favorite examples is the one with wolves and dogs. So as you probably all know, dogs are domesticated wolves. So this domestication took place around 40,000 years ago, and they each went on their own evolutionary track. And in dogs, you see there this gray line. So this represents selection for a black coat color. So at some point, there was a mutation that gave some dogs a black color. People liked this and started selecting for it, and it increased in frequency. And later on, dogs and wolves hybridized, and this gene for the black coat color jumped from dogs into wolves. And suddenly you had wolves that were also very dark. So you can see a nice example here. And it turns out that this dark coat color was actually beneficial for hunting, because when they are hunting in the forest, they are better camouflaged. So it turns out that there was selection for the black coat color in wolves, and it became a more dominant form in the US. So you also see this in birds. So here you have the Darwin's finches, which are known for their beak morphology. And it turns out that the genes involved in this beak morphology, they have been exchanged between different species. So the very rapid evolution you see in some populations can be explained by the exchange of genes related to beak morphology. And another nice example is saltmarsh sparrow and Nelson sparrow in the US. So as you can see from the name, the saltmarsh sparrow lives in the salt marshes, very salty environments. And the Nelson sparrow has been able to colonize new environments by getting genes from the saltmarsh sparrow. And it adapted to the salty conditions and increased its range. And another consequence of hybridization could be hybrid speciation, where you get a new species arising. So I put it as 1 + 1 = 3. And the best known example in birds is the Italian sparrow. So in blue, you see the house sparrow, which is quite common. And then in Southern Europe, you have the Spanish sparrow in red. And at some point in the past, they hybridized, and this hybrid population got isolated in Italy, giving rise to the Italian sparrow. And you can see that here. So if you look at the genetics, this is house sparrow, this is Spanish sparrow, and the Italian sparrow is right in the middle. And if you then look at the genome, so every bar here is an individual. So all the house sparrows are 100% house sparrow, all the Spanish sparrows 100% Spanish, and the Italian has roughly 60% house sparrow DNA and 40% Spanish sparrow DNA. So it's really intermediate. So now that you have these concepts, so especially introgression and hybrid speciation in mind, we can dive into my study system, the true geese. And when I started my PhD already in 2012, I noticed there was no phylogeny. So it wasn't known how species are related to one another. And this is one example. So here you have the lesser white-fronted goose. And some studies put it together with the greater white-fronted goose, which you would expect if you look at morphology. But there were some other studies that clustered it with pink-footed goose and Taiga bean goose. And this is just one example of the inconsistencies between different studies. Now we know there's extensive hybridization between these species, between different goose species. So I put 2 and 2 together and thought, well, what could be the impact of hybridization on the evolutionary history of geese? So could it be that hybridization explains our failure to resolve the phylogeny? So to do this, I started collecting blood samples of different species. So here you see me holding a gray-leg goose and one of my colleagues taking the blood sample. This is a friend of mine holding a Ross goose. This is a red-breasted goose, very beautiful bird, and then the big Canada goose. So in the end, we managed to sample all the species. So we had 19 goose genomes, and you can roughly divide them into the gray geese, the white geese, and the black geese. So now we have the blood samples, we get DNA from it, we sequence it. If you want to do phylogenetic analysis on genomic data, there's 2 main approaches. One approach is to take all the genes that you sequenced and you paste one after the other, and you make a kind of super gene. And then you analyze this sequence as if it is one gene, and you get a phylogeny out of it. So this approach, also called concatenation, has been criticized because you assume that all the genes have the same evolutionary history, which is often not the case. So that's why most people prefer the so-called super tree approach, where you make a gene tree for each gene, and then using different algorithms, you combine these gene trees into a species tree. And what you then often see is that different genes tell different stories. And this is nicely illustrated here. So let's assume we have 3 species, a yellow one, a red one, and a blue one. And if you look at the gene trees, it turns out that 45% clusters red with blue, 43% yellow with red, and then the remainder is yellow with blue. So different genes, different stories. And there's several explanations for this. One of them is so-called incomplete lineage sorting, which is the process that when you get the formation of new species, the genetic variants get sorted into these different species. And this sorting is kind of a chance process. So by chance, a variant can end up in the wrong species. So it doesn't follow the species tree, but it has its own evolutionary history. And the other one, the other mechanism that I'm most interested in is hybridization. So if there's hybridization between species, one gene can jump from one species into the other. And when you then make a gene tree, it will deviate from the species tree. So I used these 2 approaches on the geese. And I was very happy to see that they both gave the same result. So on top, you have the genus Branta, and below you have the genus Anser. Now this looks a bit boring, so that's why we decided to make these figures. So you get a nice overview of the relationships and the distribution. So this is the genus Branta, and you can see that the first one to split off about 7 million years ago is the brent goose. And later on, about 6 million years ago, you get red-breasted goose, and then there's a complex so-called white-cheeked geese, because the cheek is very white. You get the Hawaiian goose, Canada goose, cackling goose, and barnacle goose. And what's interesting is that Canada goose and cackling goose used to be considered one species, but it turns out that cackling goose is actually more closely related to the barnacle goose. So what probably happened is that a population of cackling goose got isolated in Europe or in Greenland, and over time it developed into the barnacle goose as we now know it. And then you have the genus Anser. And what you see here is that the first one to split off is the bar-headed goose. So that's the one that migrates over the Himalayas. Then there's a big split between North America, where you have the white geese, that's emperor goose, snow goose, and Ross goose. And then the gray geese with the swan goose, the gray-leg goose, and then the white-fronted geese and the bean goose complex. And I'll come back to the bean goose complex later on. And you can also notice that the speciation times here are very close together. So it was one speciation event after the other. And this probably explains why it has been so difficult to resolve this phylogeny. So now that we have the phylogeny established, we can start looking for gene flow for introgression. And one of the most commonly used statistics is the D-statistic. So it was developed to detect gene flow between humans and Neanderthals, as you can see here. And it's actually quite simple. You look for two patterns in the genome, ABBA and BABA, and then you calculate this statistic. And I'll walk you through it. So first you have a phylogeny with 3 species and an outgroup. So for example, here you have the barnacle goose, the cackling goose, the Canada goose. And as an outgroup, you can use one of the Anser species, like the gray-leg goose. And then you start checking every position in the genome, and you look for a pattern where species 1 and the outgroup have the same variant, and species 2 and species 3 have the same variant. So that's your ABBA pattern. And for BABA, species 1 and species 3 have the same variant. So I did this for the geese, and I found about almost 19,000 ABBA and 16,000 BABA patterns. You put this in the formula, and this resulted in a D-statistic of 0.087 and a Z-score of 4.3. And how should you interpret this? Well, if your D-statistic is different from 0 and your Z-score is bigger than 3, then it indicates there might have been introgression. And the Z-score is kind of a resampling approach, and that is used to calculate the kind of p-value that you can use. So in this case, the D-statistic is different from 0, and your Z-score is bigger than 3. So this indicates hybridization or introgression between cackling goose and Canada goose. And you can see this in these patterns because there were more ABBA patterns, and ABBA indicates that P2 and P3 are sharing a lot of genetic variation. So I did this for all combinations of species. And this is just a quick overview where you see all these arrows indicate significant D-statistics pointing to introgression, including a quite surprising one between Hawaiian goose and red-breasted goose. But I'll explain this later on. And I did the same for the genus Anser. So here as well, a lot of significant D-statistics pointing to a lot of introgression between different species. So if you put this together, you can say, well, the tree we made is so influenced by introgression that it's not a phylogenetic tree, but more of a network. So you can see that here with a very complicated structure indicating gene flow between different species. And it's more complicated for Anser than for Branta, which was also reflected in the D-statistics. This idea that evolution is not really a tree, but more of a network. You also see it in other groups of animals. So here, for example, you have different wolves and their relatives, and you see all these interconnections between different lineages. It has also been found in cows, the famous Heliconius butterflies. Here you see baboons, and even in whales, you see a network at the center of their evolution. So it seems to be quite common since we have genomic data available. But one thing to keep in mind is the idea of ghost introgression, because as you can see from these networks, for example, there was a special one between Hawaiian goose and red-breasted goose. So it could also be that it's not these two species that exchange DNA, but there's an extinct lineage that was kind of an intermediate in the gene flow from one species to the other. And this was nicely shown in these leaf warblers. So when you look at their nuclear DNA, they are quite similar. But if you then sequence their mitochondrial DNA, they're very different. And this very divergent mitochondrial DNA probably came from an extinct species that hybridized with, in this case, the blue species. So it looks as if they're very divergent, but this can be explained by this extinct lineage. So what I think is that a lot of these significant D statistics in geese can be explained by ghost introgression. So I wouldn't say there has been gene flow from red-breasted goose to Hawaiian goose. It's probably some ghost lineages in between. So I've drawn them randomly here on the network because you don't really know where they are situated. But it's something to keep in mind, and new techniques are being developed to take into account this ghost introgression. Another thing that came out as a surprise was the idea that the red-breasted goose could be a hybrid species. So I made these hybridization networks. So it's a software that takes gene trees and then makes a network out of it. And one of the solutions that it came up with was the red-breasted goose as a hybrid species between the Brent goose and the ancestor of this whole clade. It could be possible, but I'm a bit skeptical. I think it's more likely that the red-breasted goose originated through classical speciation, but then hybridized with the ancestor of this group and also hybridized with the Brent goose, which gives the impression that it's a hybrid species, but it just picked up DNA from different species along the way. So that was a big overview of the evolution of geese. And now I want to take a step back and focus on just one particular group of species, the Taiga bean goose and the Tundra bean goose. Because during my PhD, I found that introgression is common, but I couldn't really pinpoint when it happened. So during my postdoc in Sweden, I decided to focus on a particular species and find out when introgression occurred. And we decided to go for these bean geese because they look quite similar, but there are subtle differences in their beak morphology. So here you see on top male birds and below the female birds. This is their weight, the wing length, and the beak length. And you can see that in gray, that's the Taiga bean goose, is always a bit bigger than the Tundra bean goose, which you can see in black. So they're morphologically, they're a bit different, but still there's quite some overlap, indicating that maybe there's also a hybridization going on. They have different breeding sites and different migration routes. So the Tundra bean goose breeds closer to Europe, whereas the Taiga bean goose you can find more on the Russian side of the Ural Mountains. And when they migrate, the Tundra bean goose prefers to migrate over mainland Europe, whereas the Taiga bean goose goes over Scandinavia. But again, you can see there's some overlap in migration routes, some overlap in breeding sites, so they're not completely separated. So during my research, we collected samples, mainly from Finland, during migration of different Tundra and Taiga bean geese, and we sequenced the genomes of 9 individuals for each species. And you can see genetically, you can nicely separate them into 2 groups. You see again this mixture plot that I also showed with the sparrows. So you have 100% Tundra bean goose, 100% Taiga bean goose. So they seem to be genetically quite different. But then we wanted to know, well, did they exchange some DNA in the past? And to do this, we used a modeling approach where we modeled different scenarios. So the scenario you see here is an ancestral population that's split into the Taiga bean goose and the Tundra bean goose, and then they hybridized during their whole evolutionary history. And it indicates that hybridization or introgression went in both directions equally. It's also possible that it was biased, so it went more, for example, from Taiga into Tundra bean goose. And then we had some other scenarios where hybridization was mostly occurring at the beginning of the speciation event, or maybe that they had secondary contact, so they were isolated for some time and then came back together. So if I give this talk live, I always ask the audience which one do you think is the most likely scenario? But here I'll give you the answer right away. And it turns out that this was the most likely scenario, which you can see here. So this is what the data looked like. So you have from 0 to 9 individuals, and the colors represent the sharing of genetic variants between the species. And when we then made the model, you can see that the distribution nicely fits the one of the data. And these models are done in coalescent units, and you can convert them into absolute numbers. And when we did that, it turned out that Taiga and Tundra Bean Geese separated about 2.5 million years ago, but they started hybridizing again roughly 60,000 years ago. So that's the time here when they established contact. And personally, I think that we only captured the last introgression event. So probably they have hybridized several times in the past, kind of like a river that goes apart, comes back together, separates again. And with this analysis and with this data, we could only capture the latest hybridization event. Now, what does this mean for their genomes? So what we did here is we looked at different chromosomes. So this is chromosome 1, chromosome 2, chromosome 3, and the Z chromosome. So one of the sex chromosomes. And we calculated a statistic called FST. And FST indicates how different two species are. So if it's 0, they're the same. If FST is 1, they're very different. And we calculate this across the genome. And then you can see there's some parts of the genome where FST is a bit bigger, so where they are quite different. But actually, most of the genome is quite similar. So we think that this introgression is homogenizing the genome. There are some parts in the genome that are resistant against introgression, perhaps because they contain genes that are related to reproductive isolation. But that's something to look into in more detail. But overall, the genome seems to be homogenized by this introgression. And it could be that they're actually merging into one species again. And if there's bird watchers in the audience, they will probably come with the question, well, what about the pink-footed goose? Because the pink-footed goose is also part of this bean goose complex. And I'm currently finishing a manuscript on this complex. And what we found is that the pink-footed goose, which you can see here, is actually embedded within the Taiga bean goose and the Tundra bean goose. So you can see it's most closely related to Taiga bean goose. So the scenario we have in mind is that there was a split first between Tundra bean goose and Taiga bean goose about 2.5 million years ago. And then later on, a population of Taiga bean geese got isolated and evolved into the pink-footed goose. But then hybridization started between Tundra and Taiga. So these two are homogenizing each other, while this one remains isolated in Iceland and Greenland. So it doesn't really interact with the other species. So it could be that this one remains and these two will merge into one species. But this is something that we're currently looking into. And with that, I'll give you the quick summary of the presentation. So in the beginning, I showed you that hybridization is quite common on a species level, but rare on an individual level. And I hope I convinced you that the evolutionary history of geese has been heavily influenced by introgression, perhaps even ghost introgression. But the impact of these hybridization events on the genomes remains to be determined in more detail. So that's some of my ongoing research. So I thank you for listening. And if there's any questions, I'll be happy to take them.
Thank you, Jente, for that nice talk. I would like to remember they can also write their questions in the chat menu and we will read it from them, or they can feel free to ask them on microphone. So does anyone have any questions?
Jente, this is Mark Haber. Thanks so much. I really enjoy reading your posts on Twitter and your notes about all the updates. I get all my hybridization news from you, so thanks for that. My question has to do with how brood parasitism and hybridization interact with each other, because in ducks, it's only the males that are imprinting on the females and not the other way around. As far as my understanding is, the females make decisions about mate choice based on who courts them versus the males imprint on the mother. But in the geese, it's both sexes that imprint on the parents, right? Because it's obligate by parental care. And so do you think the genomic consequences of this directional imprinting, could be traced in the hybridization patterns of the genomes themselves?
That's very interesting. I think, like you said, in geese, it's biparental. And I think what you can do is look at introgression patterns of different parts of the genome. Mm-hmm. Because, for example, mitochondrial DNA is only passed on to the female line. So you could compare introgression of mitochondrial DNA with the autosomes, or you can look at the sex chromosomes. Sure. And based on the patterns you expect from mate choice and imprinting, you would expect certain patterns in the introgression patterns. Yeah. So
it'd be really fun to explore.
Yeah, something to look into, because it would be fun to extend this beyond the ducks and the geese and see how it goes in other species as well.
For sure, for sure.
Yeah,
thank you.
Interesting.
Thank you. We have a question from Zoltán Németh in the chat. Thanks for the great talk. Are there any life history traits that are particularly associated with a higher likelihood of hybridization?
So I know there's a study by Christoph Rendler from beginning of the 2000s, and he did some analysis of different life history traits and see if they could predict hybridization. I don't remember all of them, but I remember sexual dimorphism played a role. Also, I have to think hard if I can remember them. I'll probably have to look them up again, but there are some life history traits in certain species that predict more hybridization in certain species. But the main one, if I remember correctly, was the fact that they have to be sympatric or parapatric. So I think if they're sympatric, the chances are lower, because then they kind of know each other and they know how to avoid hybridization. If they're parapatric, they occasionally meet each other and they haven't evolved differences well enough to discriminate one another. So you get more hybridization in parapatric species pairs than in sympatric ones. So that's the main one that I remember at this point.
Okay, thank you. That really makes sense. We have a question regarding those sparrows and in general hybridization afterwards. So if the Spanish sparrow is present in only 40% in the genome of the Italian sparrow, and the house sparrow has like 60% of the genes involved, then would it be expected that the introgression rate is different with the parental species afterwards, if these hybrids hybridize with the parent? Or is there any kind of difference in success in this case?
Yeah, that could be the case. So I know there's some hybrid zones in the Alps between, I think it's mostly Italian sparrow and house sparrow. And you would expect that because it has less DNA from the Spanish sparrow, it's probably, there are some incompatibilities there. So introgression between Italian sparrow and Spanish sparrow will be lower. Mm-hmm. And I think they are looking into this now. So I just wrote a blog post about this, where they showed that it turns out that different combinations of house sparrow and Spanish sparrow never occur in the Italian sparrow. So they are incompatible there. And this could then have a consequence when they again hybridize with one of the parental species. This combination can pop up and lead to inviable or sterile hybrids. So it would be interesting to look into how the makeup of the genome influences introgression dynamics at this point. Yeah. Mm-hmm. It's not my study system, so I don't know all the details about how this works. but it's definitely very interesting to explore.
Thank you. Does anyone have any other questions?
Yeah, I guess I have another question, which is the bird watcher in me is when do we talk, about species collapse? What's going to be your criterion for, turning the Tundra geese or the Bean Geese into one?
Yeah, that's a very good question. So I always avoid it in the presentation, but it always pops up in the questions. So I think you'll have to look at the genome to see, because we found that it seems to be homogenizing. So it seems to become more similar over time. But you have like these small islands of differentiation, so small genomic regions where they're different. And if they are kind of strong enough to resist this introgression, it will prevent species collapse. But once you see these islands also disappearing, then you're going towards species collapse. Yeah. But it's a very gradual process, so it's going to be very, very difficult to draw a line. Like now they're different species, and then the next day they're the same. So I think taxonomists will have a lot of fun figuring out where to draw the line.
That's good. Sounds good. Thank you.
Know a bunch of people who would be really sad if the two species would be lumped.
Yeah, I don't see it happening, honestly. I think the genomic islands are strong enough to keep them separate.
They have a different migration patterns, so this could conserve them, yeah?
Yeah, so it could be that, I don't know if they looked at hybrids, if they have different migration routes. Because in songbirds, it's a large part genetic. So you get hybrids that migrate intermediate, and then they end up in places where they cannot survive. But in geese, the migration is cultural, so it's passed down from parents to offspring.
Mm-hmm.
So it could be that hybrids don't really suffer from a different migration route. They just follow their parents.
I see. I have a question related somehow to Mark's question on this. So a bunch of, let's say, rare species here in Hungary or anywhere in Eastern Europe pop up in mixed flocks of other species. So I know that's not good. A barnacle goose, if it comes down with some white-fronted goose and something like that, they spend the winter here, and afterwards they something, they go together back to the breeding grounds, the barnacle goose goes with them. Does this mean that it would be a proper chance to have some hybrids afterwards, or they maybe just separate somehow?
That's a good question. So we know that mate choice happens on the wintering grounds. So if you see a barnacle goose migrating with white-fronted geese, it probably mated with one of them, or it's just following them. Mm-hmm. But of course, also as Mark mentioned, the female has to accept the male.
I see.
It could be that a barnacle goose is imprinted on the white-fronted geese, but it still has to find a partner that's also willing to mate. So when they migrate back together to the breeding grounds, there is the chance that you will get hybrids, but it's very difficult to tell. You would really have to go there and observe the breeding population.
I see. Thank you. Do we have any other questions? If not, I wish to thank Jente for his great presentation. Thank you. Mark is clapping, so I guess everyone else is clapping virtually. It was nice to have you here. Next week's guest is Professor Nick Lane, so I will hope to see you all here also. And have a nice day and have a nice weekend afterwards. Bye.
Thanks. Bye-bye.